All right, what we're going to be doing here today, I'm just going to go over the lab that you guys are going to be doing while I'm gone. Some of the stuff should be review, but you really need to take the time to put the effort in. More than likely, this will be a lab report that you'll write after break. So if not, the explanation tool, I'm not too happy with how some of you are doing on those, so maybe we'll just go back to a lab report. So it should take you Thursday and Friday. I gave Mr. Zenchek specific instructions on what needs to be done each day. A couple things we're going to do. This is from an IB program, a National Baccalaureate, so I'm just going to cross off some of these things. And I'm going to go through, there are certain things you're not going to have to do, but we're going to do time of the cell cycle lab. You're going to be observing every cell in 2 images of onion root tip. Now, I'm going to scroll down here. You're going to get 2 pictures of onion root tip, this being one of them, and this being the second. Now, what you're going to have to do, and I'm going to try to highlight this, you're going to have to go through and count. Now, as you're going to see, it says count every cell. So this is one cell. You get the idea. I'm going to highlight several of these cells here. My lines aren't very straight. But you need to go through and make a data table of the different phases: interphase, prophase, metaphase, telophase, telophase, and cytokinesis. Telophase and cytokinesis can kind of go together. Now, you're going to get to some points where you can't tell, like, what is this cell? That's up to you if you count it or not. So if you get to a point and you get frustrated and you're like, I don't know what it is, just skip it. You may want to check with the people around you, though, because all the groups will have the same picture. So you're going to go through, and the easiest way to do this is just have 2 people working together and one person just go down through each image and counting up the what each cell's in. You may need your book to review. So where it says data collection, which is, I'm trying to get my pen here, excuse me. This is the data collection. There you go. Observe every cell in 2 images. There's your phases, the best done in pairs, record result at IB. We're not doing International Baccalaureate. We'll do AP quality data collection table. So making sure you keep control of keep a good data table there. Go down. Speaking of data tables, a couple quick things. Mr. Zenchek can explain this. Convert the number of cells in each cell cycle to percentage. Hopefully it'll come up to 100%. Then generally onion root tips take about 24 hours. And so it's approximation. So what you want to do is you want to keep this on the base of a 24-hour cycle. Try to figure out the amount of time in hours and minutes. Now here's a hint. There's a common mistake. For example, when you're figuring out, many people would incorrectly say 10.3 hours is actually 10 hours and 33 minutes when actually it's 10 hours and 60 minutes. 0.33, so 3/10 or a third, excuse me, of an hour would be about 20 minutes. So try to get that as close as possible. You need to show at least one example calculation and one worked percentage of cells and one time per stage. So I need to see just 2 of those, percentage and time. Like I said, if I decide to have you guys do a formal lab report over Christmas break, you're going to have to show some calculations. And then again, where it says create an IB appropriate quality graph, we'll keep it AP quality graph. So make sure it displays the time 24-hour cycle. And that's up to you on what kind of graph it is. Questions you need to do are right here. Just using a piece of paper for now. Which stage is shortest? Does this comply with what you know about chromosomal events during the stage and why? Going back to what occurs in interphase, and this should be review. Which stage seems to be longest? Does this comply with the information you receive about the events during the stage? And if your observation had not been restricted to the area of the root tip, how would your results have been different? So again, you're only looking at where the root tip is dividing. You're not looking at mature. Where this says FDQ, those would be like our FRQs due Friday. We're just going to kind of cross that out. So there's that first part. There's your images. This should all be Thursday. So I told him that you should have your data collected and you should have a data table. Thursday starting Friday, this is where you're going to do your chi-square analysis. Now this is actually from the AP Bio lab manual. So I've found in the past that growing the roots is not viable. It just doesn't work out too well. So what we're going to do is going to kind of simulate it. So here's our experiment. Scallions, which are a type of onion, were treated as described in the AP Biology lab manual. We had our treatment groups. We had a control, which was water, lectin, and some caffeine at different concentrations of caffeine. The scallion root tips were growing for 48 hours in the dark. Again, we're looking at mitosis. They were fixed, which means mitosis was stopped They were stored, and here's the results observed at 400 power. Again, I've tried this lab in the past. It does not go over well. Here's your control. At interphase, there was 460, prophase, metaphase, anaphase. So here's your total number. There's the lectin, caffeine, 5, the different concentrations, 10. At the 100, it was cytotoxic. Nothing occurred. So, the cells didn't even, the cells died. What you need to do is go through, you're not going to be graphing anything just yet, but what you just did previously in figuring out the percent in each phase of the cell cycle, you're going to do the same here using this data, but you're going to be creating a chi-square for each one. So let me just kind of show you. So this is just showing you the process of how these alternative treatments, like in the caffeine and lectin, were done. They were heated in 1 molar hydrochloric acid, which is pretty strong, which I would never have you guys do in the lab. That's one of the reasons why we don't do in class, for about 5 minutes. Rinse it, we stain them, blot them, you add some water, use needles gently tease apart root, and you squash the tip. This is, in my opinion, not a very good process, and I haven't found anybody that actually likes this method here. So, and it was dyed. This is what I want you to do, though. This is where you need to figure out your data. I want you to do the chi-square test for treated roots. Now, so in the control, 86% of cells were in interphase, 14% were in mitosis. This should be, if you haven't figured it out yet, this data here you're going to collect, that'll be your expected. So, sorry about that E. As you're going through doing your different chi-squares, 86% of the cells should have been in interphase and 14% of mitosis, bearing no extenuating circumstances and confounding variables and other good stuff. Calculate the expected numbers of cells by multiplying the total. So you're going to calculate these based on the 86 and the 14%. The observed, and you're not going to like this, you're going to have to flip back. And so what I'll do is I will go through and maybe photocopy this. This is your observed. Okay? This is the data you're going to use to fill out these charts here. So observe, expected, so on and so forth. What you didn't see on your paper, and I don't know why, this is where you put your chi-square data, right there. So when you do your totals, for whatever reason it didn't come off on my copy for you. So that's where you put your chi-square. What I want you to do, though, before you do any of this, which I don't have written anywhere, I really need you to come up, hopefully you remember, let me get my pen here. You need to come up with your null hypothesis and then the other hypothesis, your prediction. So if, or the scallions treated with lectin would have no statistical difference, and it would be your null, and then your real hypothesis that there would be a statistical difference. Because what you're going to do from the chi-square data that you collect there, you're going to go, and really all I want you to do is you're going to accept or reject null hypothesis, use a 5% probability. And here is the chart looking at our 5% right here. You can figure out degrees of freedom and all that. You are just collecting the data at this point because I cannot, for whatever reason, you guys refuse to do your explanation tools properly if I'm not there to hover over you. So, again, what you need to get done is for Thursday, you need to at least collect the data and in a data table. Mr. Zenchek's going to check it. If you don't have it done, you get your 0 out of 10. If you screw around, which some of you can't be mature enough to do your work. Friday, you need to have all of the data collected, all data. You need your chi-square done. I need to see about your null hypothesis being accepted or rejected. Okay? Get those two things done and you'll be good. And then there is another activity I have planned. So that's all you have to do. So hopefully you can handle that on your own. I'll be checking in on you once or twice over the course of the week. So again, good luck. This should not be that difficult. So please just do what you're told. And that's it. Good luck.